Welcome to part 2 of chapter 3 for EFB103. All right, so we've talked about the first two main macromolecules, nucleic acids and carbohydrates. Let's talk about the last two now, starting with protein, which is our third kind of macromolecule that can form these long chains like we've talked about with the other two. So proteins are incredibly diverse. You can read up on this in the textbook, but here are just some examples and you get a sense of it. We're trying to squeeze all these different functions, and these are just a subset of the functions that proteins can do into this one table here. And hopefully just by looking at this, you appreciate the diversity and really flexibility of proteins. And we'll see that throughout the course. Proteins are like the doers. They're structural, they're enzymes. That doesn't mean the other macromolecules aren't involved in those things as well, but these guys are just very, very diverse. When we think about that field of proteomics, which I mentioned last time, the study of all the proteins in a living thing or in a cell, that is a much more complicated thing than, say, genomics, looking at the genome, because of this diversity of function. And we'll see how this arises as well when we get into gene expression. So proteins are made out of amino acids. That is our building block monomer unit. There are 20 different types of amino acids. There are some subtle variations on that in some groups of organisms, but for the most part, we can say there's 20 types of amino acids, and they all have this very similar structure. You have this central carbon, which is called the alpha carbon, a hydrogen here, an amino group, a carboxyl group, and then this R group. Now, R doesn't represent an element. It represents a variable site. So this is where we see differences in amino acids, and we'll see that in a moment. But first, all right, so yeah, it's the carboxyl group that makes this an acid. This is an amino acid, so carboxyl or carboxylic acid group here. All right, so what do we see with those variable R groups? Here's the baseline amino acid highlighted in purple for all of these. And the differences here are at the top here. This is that variable R group, and we can categorize amino acids based on their properties. So these ones up here that have the R groups highlighted in yellow, these are the non-polar or hydrophobic amino acids. These guys in green, these are the polar or hydrophilic amino acids based on these R groups. And these guys are also at the bottom here, hydrophilic as well, but they form ions, so as the acidic and basic amino acids. Now, obviously, these things are going to determine the properties of a protein. And they're going to be located, different kinds of amino acids may be located in different places in a protein. And so a question for you might be, where would you expect to find, say, the hydrophobic amino acids versus hydrophilic amino acids? So think about that for a second and answer this next question. All right, so in a globular protein in an aqueous solution, say water or cytoplasm, you're going to find those hydrophilic amino acids on the outside interacting with water, and then buried inside that protein, you'll have the hydrophobic amino acids. Another time we see hydrophobic amino acids as part of a larger protein is transmembrane proteins. We'll get into that here as we get into cell membranes. You see the hydrophilic stuff on the inside and outside of the cell interacting with water, and then what's in the middle are the hydrophobic ones. So let's start putting these together now and building a protein. When we can start connecting amino acids, what we create is something called a polypeptide. This is a linear chain, a non-branching chain of amino acids. And that creates the primary structure of a protein. We'll get into that. Now a protein, it is sort of the finished product of a protein might actually consist of more than one polypeptide, whether that's because multiple polypeptides come together or a polypeptide's produced and then it's cut and then those pieces come together. But a protein can be built out of more than one polypeptide. Not always, sometimes it is just one, but it can be made of more than one. So amino acids are joined by dehydration synthesis, and what that bond that forms between them is called a peptide bond. So go back to the first bullet here, polypeptide, that's what we're talking about here. So we're building these chains, one amino acid at a time. What's connecting them? A peptide bond. And so here's what that looks like. Here's that dehydration synthesis. That's our chain building. We've seen this before. Out comes water, that's dehydration. We create a covalent bond. So a peptide bond is a covalent bond. It's just, we just give it a special name because it occurs between these amino acids here, but it's just a carbon-nitrogen covalent bond. And that's how we build proteins, polypeptides. So we start putting these together, that builds this chain of amino acids. But what really matters is how that protein, that polypeptide folds and forms the shape and therefore determines the function of that protein. And here's kind of a cool example. I just love this image. So what you're seeing here on the left is the very end of an antibody molecule. So it says antibody protein, but it's really the kind of the tip of the antibody that interacts with pathogens or pieces of pathogens. And then you see a protein from flu virus. And look, even just looking at this, you can see that these match up really well. So that interaction, the shape, the charges, all that is critical for how this antibody works with this flu protein. So the structure really important for the function. So the structure of a protein determines its function. That string of amino acids we've already talked about, that's called the primary structure. Most proteins, that's how they're initially produced, but they will start folding and form their final shape in terms of how they function. Some might not do all that much folding and don't look too much different from the primary structure, but most of the time these are folding up and we're sort of building on to levels of structure here. The secondary structure, we start to see that protein folding and twisting. Tertiary, we start to see those pieces now folding on each other. And then finally, quaternary structure, we see multiple polypeptide subunits coming together to form a final protein. So here's that primary structure. It's, again, just that string of amino acids. No foldings happened yet. This is what you would imagine theoretically is being produced as you're making a protein. In reality, that protein, even as it's being made, starts to fold on itself. Secondary structure, the elements that we see commonly in secondary structure are these two things. Our alpha helices, so imagine, the old style telephone cord, it's just a coil, and a beta sheet. And this is that protein, that primary structure zigzagging back and forth. You see the little arrows here showing the orientation. And what's connecting either the helix or the sheet together? These little dotted lines, these are hydrogen bonds. So hydrogen bonds are super important at this secondary structural level, forming these types of common structures that we see within proteins. In tertiary structure, third degree structure, now we start to see those helices and sheets folding on one another, or some of these other interactions. So maybe, I mentioned before, you might have hydrophobic amino acids that are going to kind of come together on the inside of a protein. So they're being sort of excluded by water that's around them and sort of pushed to the inside, and that's going to cause that protein to form a certain shape. Other things that we might see, so here's that hydrophobic interaction, ionic bond here. So you've got two charged amino acids and they're being attracted to one another, just like magnets like we saw with ionic bonding. Hydrogen bonding still occurs as well in tertiary structure. Again, it's more important at that secondary structure level, but it's, doesn't mean it's not important here. Another kind of bond we see in tertiary structure are disulfide bridges or linkages here. So cysteine is one of our amino acids, and it contains sulfur. And you can end up with a covalent bond between the sulfurs of two cysteine amino acids on this polypeptide chain. So let's fold this up even more to end up with that final tertiary structure. For some proteins, that might be the end of it. But for others, they may need to come together with other subunits or other polypeptides to form that final functional protein. In that case, those proteins would have quaternary structure if they're made out of multiple polypeptides. Again, some are not. A nice example of a protein that is made out of multiple subunits is hemoglobin. So really important protein in your body, hemoglobin is, and it is made out of 4 subunits, alpha subunit and beta subunits, those 2 of each of those. And then you have this heme group and iron. That's important for moving gases around in your blood. So take a look at this structure. What is a common secondary structural element that you see in hemoglobin? It's everywhere if you look at this picture. All right, so besides that amino acid sequence and the properties of those amino acids, there's other things that can determine the structure of a protein. So pH, salt concentration, temperature, other proteins that are present, other, say, environmental factors as well. So there's a whole bunch of things that can determine that shape of a protein. And in this little example here, this cartoon showing this protein at 2 different pHs, low pH and high pH, you can see, if you sort of focus on these alpha helices here, when you increase the pH, the protein, it almost sort of flips. So you've got these beta sheets over here instead of over here. And it has a different function at those different pHs. Sometimes a change in temperature or pH just basically unfolds the protein, makes it non-functional, and that loss of structure is called denaturation. So you basically inactivate that protein. That can be shown here as well. Denaturation by whatever means. Let's say temperature, we heat this thing up, it unravels, all that folding that has happened when it was formed kind of gets undone. You're breaking those hydrogen bonds. You wouldn't break disulfide bonds unless you treated it with a chemical that would do that, but just heating would definitely disrupt those hydrogen bonds and some of those other interactions, and you end up with this denatured non-functional protein. Sometimes this is reversible, so let's reduce the temperature, and this thing will fold back up because it folds because of the properties of its amino acids. Those are still there, and it's going to reform this shape. A couple of other terms, just a little bonus information here that you might hear are motifs and domains. A motif in a protein is a common element that we see. So these are a couple of examples. A beta alpha betix beta motif. So you have a beta sheet, alpha helix, beta sheet. Helix turn helix. We see a lot of those in, say, hemoglobin, for example. So these are just common structural themes that we often see. Another term that we encounter is domain. So proteins are often assigned, regions of proteins are assigned domains or given names. And this is shown here. This is a single polypeptide. So if you follow it along, it's all connected, but it's colored differently just so we can understand this idea. And these domains or parts might have different functions. So sometimes we might say, well, that's the protein binding domain. Maybe this is a protein that sticks to other proteins, or this part of the protein crosses a cell membrane. So we'd say that's the transmembrane domain. And so these are some important terms that you'll hear. It's the one you've all been waiting for, the lipids. So this is our final group of macromolecules. and I'm going to say it probably many, many times, lipids are not just fats. So we look at the little cartoon here and we might get the impression like, oh, here's a triglyceride made out of fatty acids. Lipids are fats. Fats are a kind of lipid, but they are not the only kind. What makes lipids lipids, or what defines them, is that they are hydrophobic, that they're insoluble in water. Not that they're just insoluble, but insoluble in what, right? Water. And so hydrophobic would encapsulate that whole thing. So fats are hydrophobic, but so are some of these other compounds as well. What do we see commonly in lipids? Lots of these non-polar carbon-hydrogen bonds. So hydrocarbon molecules are often lipids. And so there are three main categories. Fats, yes, that's a kind of lipid. The phospholipids, there's some nuance there. And the steroids. So here's cholesterol in this cartoon. That's an example of a steroid. So let's start with fats. Fats are also known as triglycerides. Their structure is composed of a glycerol molecule with 3 fatty acid tails attached. These are a great energy storage molecule. So we saw that carbohydrates were a good energy storage molecule. You get about 4 calories per gram out of carbohydrates. Lipids, or fats, I should say, you get about 9 calories per gram of fat. So much even better energy storage molecule than carbohydrates. So these fatty acids, these tails, this is sort of where we get our bang for the buck in terms of energy. These are hydrocarbon chains, and they may be saturated, unsaturated, or polyunsaturated. And you may be asking, saturated with what? It's with hydrogens. So here is an example of a saturated fatty acid chain here. So here's carbon, carbon, carbon skeleton. Notice the carbons are completely saturated. There's no double bonds. There's basically as many hydrogens as you can get on here. That is a saturated fatty acid. The other thing I want to point out here is this is where we see some similarities to our other macromolecules, where we have a dehydration synthesis reaction here, where we're taking this glycerol, this is a 3-carbon molecule with these OHs on it, and we're attaching this fatty acid. Why is it an acid? Here's that carboxylic acid group. And we're losing a water, so it's dehydration. And we're attaching these fatty acid chains to this backbone. Now, this doesn't form long chains. You only get 3 fatty acid tails on each one of these. But again, that's where there is some similarity to the other macromolecules. All right, so we have these ester linkages that connect these tails to that glycerol backbone. And here is our triglyceride. So 3, 1, 2, 3, triglyceride. The saturated fatty acids that form these triglycerides, they are going to form like a pretty stacked up association with each other. And this space filling model gives you a sense of that. So they're all very tight next to each other. And the consequence of that is that if you have a bunch of these, they can squeeze in and be more dense. And if they're more dense, then they tend to be solid at room temperature. So here's the example of butter. It is solid at room temperature because it's made of all these saturated fatty acids or saturated fats. Vegetable oil, on the other hand, is not solid at room temperature. It is made out of fats that have these double bonded carbons and therefore unsaturated fatty acid chains here. So they're unsaturated because if you look, say right here, you could in theory add more hydrogens here, but instead there's a double bond. So it's unsaturated. The other thing that this double bond does is, or many of these double bonds, is they put kinks and bends in this. So now you can't squeeze as many of these fats into the same amount of space as a saturated one, and they are less dense, and therefore they're liquid at room temperature. How about trans fats? You've probably heard of these. So what trans fats are is if you take our unsaturated fats and hydrogenate them. So we're adding hydrogens back onto those, saturating them or getting close to saturating them. And so you can fully saturate some of them, but in the process, you'll also produce these trans fats. So here's a trans fat here. Notice there is still a double bonded carbon, but for the most part, this is still fairly linear. It's not all bent and curved like this unsaturated vegetable oil fat. These trans fats are associated with coronary disease. I say associated with. Not necessarily the direct cause, but there is an association with heart disease and trans fats. So are trans fats solid or liquid at room temperature? Looking at the structure here. Right, so they are much more linear like the saturated fatty acids, and therefore they're going to be more likely to be solid at room temperature. All right, another kind of lipid are the phospholipids. These guys are particularly interesting because they're made out of that glycerol backbone like we saw with fats. You have 2 fatty acid tails, not 3. And instead on that 3rd carbon of the glycerol, we have a phosphate group. So this is interesting because now you have a molecule that is nonpolar in some ways with those fatty acid tails, but you have this phosphate group. And if you remember, phosphates are super polar. So this ends up creating a molecule that is both polar in one part and nonpolar in another part. And how we describe that, the term we use to describe that is amphipathic. So it has polar and nonpolar regions. Here's what a phospholipid looks like, a couple of different ways to look at it. Again, those nonpolar tails. Here's our glycerol, nice big polar phosphate group, as well as this choline group here on here. But it's the phosphate that's really driving that polarity. So why are these so important? We'll see these again when we talk about cell membranes. Because of these 2 components, phospholipids will spontaneously cluster together. That phosphate head region interacting with water because it's polar, but those nonpolar tails really being pushed away from water. And since they're being pushed away from water, they tend to clump together. And they will first form almost like you imagine little bubbles with those tails oriented inward. These are called micelles. And eventually these will get bigger and bigger. Let me show you a picture. So here's that micelle with the tails oriented inward away from the water because they're being forced away from the water. And then this will get big enough that ultimately it will collapse and form a sheet. And that's what we see here. The head's on the outside on either side of this, and then the tails oriented away from water on the inside. And this is the basis of cell membranes. How this is described or how this works is through a process called hydrophobic exclusion. So the heads are interacting with water, the tails are being excluded. These tails are not really interacting with one another per se, it's just that they're being pushed away from water, and so they're stuck facing each other. All right, last group of lipids. These guys will come up again when we talk about cell membranes. It's the steroids. So steroid molecules typically have this 4-ring structure and then a whole bunch of other different things going on that are attached to them that determine their properties. Cholesterol is a great example of a steroid that's important in cell membranes. Many hormones and vitamins are also steroids, so we see some examples here. Again, here's that 4-ring structure. Vitamin D3 sort of almost has that 4-ring structure, but not quite. So that's where typically they have a 4-ring structure. But if you see these kinds of chemical structures, usually you're thinking hormone or vitamin, steroid, and lipid. All right, so those are our lipids and proteins. We'll see you in Chapter 4.